At the very heart of life itself, there's this quiet, constant process of renewal. It's this perfectly controlled system, an incredibly intricate dance of molecules that's absolutely essential for our existence. Today, we're going to pull back the curtain on this amazing process. I mean, really think about it. You get a paper cut, and without you even thinking about it, your body just springs into action. A few days go by, new skin forms, and poof, the cut is gone. It feels like magic, right? But it's actually biology at its most precise and powerful.
So that healing, it's all thanks to cell division. When you get a cut, the healthy cells around that injury get a signal to start multiplying. They create perfect copies of themselves to patch up the damage. This super organized process is critical, not just for fixing injuries, but for the basic day-to-day maintenance of our bodies.
But here's the thing. With all that incredible power for order, there's also this huge risk. Because if that control gets lost, if even one tiny step in this carefully choreographed dance goes wrong, the result isn't healing, it's chaos. And that chaos has a name we all know: cancer. Okay, so how on earth does the body manage this incredibly high-stakes balancing act? Well, it follows a very precise instruction manual, a biological blueprint for growth and division. And that blueprint is called the cell cycle. So, the cell cycle, you can think of it as the life story of a single cell, from the moment it's born to the moment it divides into two new daughter cells. It's a continuous loop, a journey that's broken down into 4 main phases. Let's do a quick walkthrough. First up is the G1, or gap 1 phase. This is where the cell bulks up and gets all its resources ready. Next, we have the super important S phase. S is for synthesis, which is when the cell meticulously copies its entire set of DNA. After that, in the G2 phase, it does one last quality control check. And finally, we get to the main event, the M phase, or mitosis, where the cell splits into 2 brand new identical cells. Now, obviously, a process this complicated and this important can't just be left to chance, right? It needs constant supervision and really tight control. And this brings us to the molecular machinery, the gatekeepers that make sure everything happens in the right order and at exactly the right time. So, who are these gatekeepers? Well, the answer is pretty fascinating. It's all down to a family of proteins that work together like a molecular engine and an ignition system for the whole cell cycle. All right, first up, let's meet the engine itself. These are called cyclin-dependent kinases, or CDKs for short. These are powerful enzymes whose job is to push the cell forward. They do this by basically flipping molecular on switches on other proteins. But here's the catch. By themselves, CDKs are totally inactive. They're just an engine waiting for a key. And if the CDKs are the engine, well then, you need a key, right? And that's where the cyclins come in. These are the regulatory partners for the CDKs. And they're called cyclins because their concentration, how much of them is in the cell, literally cycles up and down. When a specific cyclin builds up to a high enough level, it binds to a CDK partner, and bam, it turns the engine on. A perfect example of how this whole partnership works is when cyclin B teams up with CDK1. Together, they form this active complex called MPF. This specific duo is like the master switch that gives the cell the final green light to go into mitosis. Okay, so it's not just one single CDK and one cyclin running the entire show. That would be way too simple. The cell actually uses different combinations, different CDK cyclin teams, to act as gatekeepers at specific points along the journey. And the system is actually incredibly specific. Think of it like different teams of specialists for different jobs. To get through that first growth phase, G1, the cell calls on the CDK4/6 and cyclin D team. Then, to handle the critical job of copying all that DNA in the S phase, a different team, CDK2 with cyclins E and A, takes over. And as we just saw, that CDK1 cyclin B team is the specialized crew that triggers the final division in mitosis. Each team has its own very specific mission. So we've got this beautiful, elegant system of molecular gatekeepers. It all sounds perfect. But what happens when it fails? This is where our story shifts from order to chaos. And it's where we find the cellular roots of cancer. Well, to keep that catastrophe from happening, the cell has these built-in safety protocols, multiple layers of them actually. The first line of defense is a series of checkpoints. These are points where the cell cycle literally hits pause, so the cell can scan its own DNA for any mistakes or damage before it gets the okay to move on. So how does this alarm actually work? When specialized sensor proteins like ATM and ATR detect something wrong, like a break in the DNA, they immediately sound the alarm. This kicks off a chain reaction that ultimately blocks those CDK engines we were talking about. It basically slams the brakes on the whole cycle, which buys the cell precious time to fix the damage.
But what happens if the damage is just too much to repair? Or worse, what if the alarm system itself is broken? That's when the cells calls in the heavy hitters, the tumor suppressor proteins.
One of the most critical ones is the retinoblastoma protein, or RB. You can literally think of it as the emergency brake for the cell cycle. When it's active, RB clamps down on the machinery the cell needs to start copying its DNA, which stops it from entering the S phase. To move forward, a CDK cyclin team has to come in and release this brake. So if something's wrong, RB stays put, and the cell isn't going anywhere.
And then there's the big one, the most famous guardian of them all, P53. It's not called the guardian of the genome for nothing. When that DNA damage alarm goes off, P53 gets activated, and its main job is to deploy an emergency stopgap, a protein called P21. P21's mission is to directly attack the CDK cyclin engines, binding to them and shutting them down, bringing the whole cell cycle to a screeching halt. This protein is so unbelievably important that in over half of all human cancers, the gene for P53 is broken, which means the cell's ultimate guardian is off duty.
So, as you can see, this whole thing is a constant battle happening inside every single one of us. It's a push and pull between the forces that drive the cell to divide and the guardians that hold it back. And our health? Well, it depends entirely on keeping that delicate balance. And this isn't some brand new idea. Way back in 1858, a physician named Rudolf Virchow understood this fundamental truth. He said that the real home of disease, including cancer, was the cell itself. Our modern understanding of CDKs, cyclins, and tumor suppressors just proves how incredibly right he was. Just think about it. Every single day, your body performs this incredible balancing act billions, literally billions of times, all to keep you healthy. We tend to focus so much on the failures, the times it goes wrong and leads to disease. But maybe the real wonder lies in the countless times the system gets it perfectly, absolutely right. What can we learn about the sheer elegance of life from its overwhelming success?